Hello, and welcome to Virtual Investor Conferences. My name is Matt Lateplo, and on behalf of OTC Markets, we're very pleased you have joined us for our next live presentation from Lisata Therapeutics, Inc. Before I introduce our speaker, please note to submit your questions in the question box to the left of the slides, and that all of today's presentations will be recorded and available for 24/7 replay. At this point, I'm very pleased to welcome Dr. David Mazzo, Chief Executive Officer of Lisata Therapeutics, Inc., which trades on NASDAQ under the symbol LSTA. Welcome, David.
Thank you, Matt. And good morning, everyone. And thank you very much for taking the time to listen to my presentation and give me the opportunity to introduce you to Lisata Therapeutics. As with all public companies, I'll just mention that I will be making forward-looking statements throughout the presentation and ask that you keep these in mind as you take investment decisions. So let's start with a very simple summary. What is Lisata? Well, Lisata is a publicly listed clinical stage therapeutics development company that has a very novel solid tumor targeting and penetration technology that's designed to improve the efficacy of anti-cancer drugs. So our product does not have any anti-cancer efficacy as a single action agent, but in fact is used in combination with existing agents or agents in development to significantly improve their efficacy while not deteriorating their safety profile. On the surface, people may ask what would set Lisata apart from other small cap biopharmaceutical or biotechnology companies, especially given that the sector has been under such pressure for at least a year. And many companies will talk to you about their seasoned management team and, of course, their proprietary and field-leading technology. But there are several things that I think highly differentiate Lisata and really make us a very attractive near-term investment opportunity. Probably first and foremost is that Lisata is not cash strapped. We have approximately $70 million reported at the end of last year, which is enough money under our development plan to take us through data on all of the programs that I will describe today, and that's through the first half of 2025, and we have no debt. So we're not in a position where we have a major financial overhang as so many other companies do. We also have a large clinical development pipeline, which, as I mentioned, is fully funded, and that provides many potential value creating opportunities for news flow and business development news flow over the course of the next 2 years. And of course, we have a platform technology that has been validated, in inverted commas, by the existence of some major partnerships already and the opportunity for a number of similar partnerships to be developed. So why oncology? Well, we all know that cancer is the leading cause of death worldwide, and that solid tumor cancers account for over 90% of all new cancer cases. And if you look at the list of cancers on the left-hand side of the slide, everything that's in the fuchsia color is a solid tumor. So you can see it's the vast majority. So what is the challenge with dealing with intratumoral exposure for solid tumor cancers? Well, the challenge is that there is a set of cells, a gamish, if you will, around the tumor called the tumor stroma that acts as an effective barrier to anti-cancer agent penetration. So if the anti-cancer agents can't get into the tumor, then they have less likelihood of being able to be efficacious in shrinking or eliminating the tumor. Additionally, the tumor microenvironment has immunosuppressive cells that contribute to tumor resistance to drugs, but also allows for metastases or the spreading of the tumor cancer cells throughout the body. And continued dosing of non-targeted anti-cancer therapy generally leads to intolerable off-target side effects, even though the logic of higher concentrations of drugs leading to better efficacy might lead you to do this. It's really limited by the safety profile of the products. So our way to address this problem is through something called the CendR platform, which is a targeted penetration technology that enhances drug delivery to solid tumor. It converts tumor stroma from barrier to conduit, allowing for much better penetration of anti-cancer treatments, and doing so in a targeted way, that is, bringing the drugs to the tumor where they need to work. We are anti-cancer agent agnostic, so we can combine our products with many different anti-cancer agents, either those that are already approved on the market or those that are in development. And the mechanism is effective with products that are either co-administered or actually chemically tethered to our lead program. Co-administration, of course, represents a streamlined path to development, and the covalently bonding or ionically bonding tethering allows for a prolonged compound exclusivity in a life cycle management type scenario. And this approach also combats resistance by selectively depleting the intratumoral immunosuppressive cells. And the platform extension can be really to all drug modalities, including chemotherapeutics, immunotherapeutics, radiotherapeutics, and even cellular therapeutics. So our lead program on this platform is called LSTA, and it is clinically advancing in a variety of difficult to treat solid tumor indications as part of our global registration strategy. Our lead indication is in metastatic pancreatic ductal adenocarcinoma, or MPDAC. And here we're combining LSTA with both existing standards of care. FOLFIRINOX, which about 50% of the patients with MPDAC receive if they are still robust enough from a general health perspective to withstand the more rigorous treatment that FOLFIRINOX provides, or the combination of gemcitabine and nab-paclitaxel or Abraxane, which is what the other half of the patients receive. LSTA has been granted a fast track and orphan drug designation by the US FDA in pancreatic cancer, and we're about to start studies in combination with these standards of care plus immunotherapies in the near term. We also have a basket trial called the Bolster trial, which is scheduled to start in the near term, and it will be looking at LSTA in combination with other standards of care applied to other solid tumors. So we're looking at a clinical development program that focuses on advancing in specific indications as well as broadly demonstrating clinical efficacy and tolerability across a wide variety of other solid tumor indications. We also have a number of partnerships that we have created, a China deal with a top 10 pharmaceutical company there called Qilu Pharmaceutical, which is developing the product in conjunction with us for the greater China market, where we receive, of course, typical milestones and royalties on sales, and where they assume all the costs for development and commercialization. And we have a number of research development collaborations, including one with Roche as part of their Morpheus program. And we expect that we would be able to create additional partnerships of this type, either regionally or with a variety of companies with different standards of care, given the general applicability of LSTA across all modalities of drugs. So let me tell you a little bit how this works specifically. LSTA in and of itself is a 9 amino acid cyclic peptide that has a very high binding affinity and selectivity for the alpha V, beta 3, and beta 5 integrins, which are upregulated significantly on tumor cells, tumor endothelial cells, and the tumor immunosuppressive microenvironment cells. So generally the area of the stroma. Once LSTA is bound to these integrins, it is susceptible to protease cleavage by naturally occurring enzymes in the microenvironment, and it's split into 2 linear fragments, one of which, the 5 amino acid fragment, is a C-end rule or CendR peptide fragment, and that fragment itself has a very high binding affinity for neuropilin-1, which is an adjacent receptor also upregulated in this same environment. And once neuropilin-1 is activated, the CendR transport pathway is activated. It's a naturally occurring transport pathway that's often used by cells for nutrient transport. But here we hijack it, if you will, and it forms these roughly 200 nanometer sized vesicles at the cell surface, which encapsulates basically anything in the circulatory system and helps it percolate through the cells all the way down into the center portion of the tumor, where the drugs are able to do their job with a much more targeted and much more concentrated effect. We also have the ability to open the gap junctions, and that allows for the body's natural immune cells to have a greater chance of penetrating the tumor and also enlisting the natural immunity toward tumor combat as well. This is just a slide showing how the showing how the molecule works in a mouse model of human pancreatic cancer in a nude mouse using the fluorescent quantum dot experiment as effectively the co-administered example of a drug. And here you can see that once you administer these quantum dots, if you apply a quenching solution, that basically kills the fluorescence of the dots everywhere in the circulation. And so if there's no LSTA1 in the body of the mouse, then you quench all the fluorescence because the quantum dots have spread everywhere. But when you administer LSTA1 concomitantly, you can see all the dots go into the solid tumor, and of course the solid tumor lights up selectively and I think very obviously. And then there are a number of publications, upwards of 200, that show that LSTA1 in combination with a wide variety of different types of anti-cancer agents in a wide variety of cancer tumor models always demonstrates an increased permeability and increased efficacy in these preclinical models. But most importantly, our clinical data from humans. And here you can see really compelling results from our phase 1b/2a trial that was conducted in first-line MPDAC patients in 3 sites in Australia, where LSTA1 was shown to be well tolerated, to have no dose-limiting toxicity safety, and that the general profile of the product from a safety perspective was consistent with the standard of care alone. This was done with concomitant administration with the gemcitabine nab-paclitaxel standard of care. And what you see here are what most people would characterize as unprecedented improvements in efficacy of that standard of care in combination with LSTA1 across really all of the critical measurements made in a cancer clinical trial, including overall survival and progression-free survival, overall response rate. So that's led us to this very broad regulatory global strategy, which I mentioned previously. And what's important about this is that we have a very capital efficient development plan where we have sharing costs based upon the partnerships that we've established, or reduced costs based upon the use of selective geographies. So I'll note a couple. The most advanced program is the ASCEND clinical trial, which is the phase 2b trial, which is being conducted in Australia, New Zealand, and Ireland. This is in first-line MPDAC with the gemcitabine nab-paclitaxel standard of care. And this program is being funded by Lisata, but what is really interesting is that the use of Australia, New Zealand, and Ireland makes this a highly economical trial, especially since we get a R&D rebate from the Australian government, which amounts to approximately 48% of all the dollars spent there. So this makes this a very, very affordable trial. We also have our basket trial, which I mentioned, which will be called Bolster, and is being conducted in a number of sites in Europe and North America. That is due to start sometime soon. And then a variety of other trials that are being done with partners who are either funding the trial entirely or who are sharing in the cost of the trial. And this is just a further list of that. And you can see that in addition to trials in MPDAC, we have trials in a variety of different solid tumors with the opportunity to generate news flow from an execution as well as data perspective over the course of the next 2 years. Now, to extend this platform to nucleic acid-based drugs, we have a preclinical program based on a concept of a tissue-penetrating nanocomplex. And here what we do is we take the SendR approach to targeting and penetrating solid tumors, and we modify that so it can be applicable to the delivery of antisense oligonucleotides and or small interfering RNAs. And the reason that this is important is that more than 95% of the ASOs and siRNA drugs get sequestered in the endosomes. And so you have this problem of stroma barrier and then also endosome sequestration. And you really have to find a way to accomplish the elimination of both of that. And our TPN platform is in fact what we think will be the way to address that. The program looks at taking a CendR targeting peptide such as LSTA1, combining it with an endosome escape moiety, and of course then having sRNA or ASOs concomitantly administered so that we form these tissue penetrating nanocomplexes, which we expect will evade endosome sequestration and allow for tumor penetration. We have a number of preclinical proof of concept experiments ongoing now, which we hope to be able to report later this year, which will demonstrate, we think for the first time, the ability to efficiently deliver this modality of drug into solid tumors, which opens up this class to a whole new application. We also, within Lisata, have a number of legacy programs, including one set based on CD34 positive cell therapy platform technology. And here we have two legacy programs that are worthy of mention. One, which is for a compound that is a trade name called Honedra, which is in Japan for critical limb ischemia at the registration stage, and another called LSTA 201, which is a phase 1 trial that's just read out in diabetic kidney disease. For Honedra, it's important to note that we have received a very special regulatory designation in Japan called a Sakigake designation. And that, coupled with the status of this program as a regenerative medicine, gives us the eligibility for early conditional approvals based upon a much more accelerated regulatory pathway to registration. We also have orphan drug designation in the US for Berger's disease and an advanced therapeutic medicinal product or ATMP designation in Europe for this program. ASO, which is not antisense or oligonucleotides, but in this case, arteriosclerosis obliterans or critical limb ischemia, is an arterial obstruction that impedes the blood flow in the lower extremities of patients with generally with diabetes, and it manifests itself as severe rest pain and non-healing ulcers. And Berger's disease is just an orphan offshoot of critical limb ischemia. These patients do not respond to surgical interventions such as angioplasty or stenting, and existing pharmacotherapy is inadequate. And so we have designed a program where we use our CD34 cells, which are pro-angiogenic. They create new microcirculation in the areas of ischemic insult to effectively promote controlled microcirculatory growth in the areas that have been deprived of blood and nutrients by the disease, and to essentially eliminate the symptoms and prevent tissue necrosis. And this is a program under this regenerative medicine law in Japan, which is eligible for conditional approval based upon a single trial that shows the trend toward efficacy, doesn't require statistical significance, and an acceptable safety profile as agreed to by the Japanese regulatory authorities. And I'm happy to report that the trial that we completed that was designed with the regulatory authorities does in fact show that there is a trend toward efficacy and acceptable safety. The treatment group reached the CLI-free status, which was the primary endpoint here, about 9 months faster than any of the standard of care groups, and the safety profile was very well tolerated. And so we are now in the consultation process with the PDMA, which is the normal process toward a Japanese new drug application. And we're also in advanced discussions with a number of Japanese pharmaceutical companies to have them license the product for potentially to complete the registration process, but certainly for the commercialization of the product in Japan. And then I'll mention LSTA 201 very quickly. Similar approach here in terms of a pro-angiogenic rationale for treating kidney disease, which is really a disease of the deterioration of the microvesicles within the kidney. And the thought process was, if we were able to regenerate kidney, then we would provide an alternative to the available therapies, which really just stop the progression of the disease or at least slow it. And so the goal of this study was to demonstrate kidney regeneration by an increase in glomerular filtration rate. And we had mixed results, but positive nevertheless, in that the chronic kidney disease patients were able to tolerate the mobilization, donation, and administration of the cell therapy, which wasn't completely obvious before we did the trial. But we did not show a consistent improvement in kidney function in all subjects. However, the lead investigator here, a very well-known clinical nephrologist, has seen a result that he thought was interesting and compelling enough that he's suggesting further clinical work. And so we're now working with a number of partners to devise a program by which we could go forward where someone else would foot the bill. So the important thing to note about these CD34 therapies is that they do not require any additional capital investment from Lisata, but they do provide potential upside for our investors. And then I'll just mention our news flow here. This is a very busy slide. It's really not meant for you to read each line, although if you download the presentation from our website, you would be able to study this. But it's really the visual impact of showing that across the entire pipeline over the course of the next 2 years, we have projected a number of operational and data milestones related to the clinical programs. And then you can overlay this with the possibility of a number of business development collaborations and partnerships should provide for a very healthy news flow and a process that we hope will lead to a continual increase in shareholder value. As I mentioned, our financial situation is very stable, having reported approximately $69 million at the end of last year with no debt and a projected cash runway going through the first half of 2025, which takes us basically through data on all of these programs, and with a very clean capital structure. And so that brings us back again to the summary of the investment rationale, which I think is worthy of repetition. What differentiates Lisata is that although we are often categorized, and certainly from a market perspective, have been put in the same bucket as so many other small cap biopharmaceutical companies, we in fact are not cash strapped. We have a very solid financial plan, a runway that takes us through data, a very capital efficient development program, in fact, a very strong history of managing capital very effectively, many milestones across the next several years. So we're not binary, and we have the opportunity for any one of those to create significant value, and the possibility of building on our existing platform of partnerships to actually create additional value, a seasoned management team, and a very healthy IP perspective. And so with that, I'll just mention that after this, if anyone has the desire for further conversation or information, in addition to going to our website, you can contact our Vice President of IR and Corporate Communications, John Mendito, and his credentials are on the screen. And at this point, I will stop and open it up for anyone who has any questions. And so the first question on the list here is, will LSTA1 have applicability to brain tumors? And I know I went very fast on that list of trials, but if you go back to the slides on the website, you'll see that we actually have a glioblastoma multiform trial that's going to be started in the second quarter or perhaps early third quarter of this year in a number of patients. That trial is going to be run at a very, very reputable set of clinical groups in Estonia, Latvia, and Lithuania, where there, for some reason, seems to be a very high prevalence of GBM. So yes, we do expect that LSTA1 will have applicability to the treatment of brain tumors, and this trial, it'll be in combination with the standard of care temozolomide. The second question is, in general, what percentage of solid tumors do you believe this may be active against? In particular for PDAC, what percentage of cases are suitable? So right now, we expect that theoretically, at least, this technology should be applicable to all solid tumors which have a stroma which presents a notable barrier to existing or developing therapies. So that's quite a large list of solid tumors, very broad applicability. In the case of PDAC, we think that this is applicable essentially to all pancreatic cancers, whether they're resectable or non-resectable, and whether the patients are receiving FOLFIRINOX or the gemcitabine Abraxane combinations of standard of care. The next one is, can you provide some color around your studies in Australia and government support? Well, yes, I can. So in Australia, we're working with a very, I would say, world-renowned cancer consortium study group called the Australasian Gastrointestinal Testing Group, or the AGITG. This is a group of sites that specialize in treating cancers of the GI tract, of which pancreatic cancer is most notable. They have a membership that spans all of Australia and affiliations in Ireland and New Zealand, and we're able to utilize their consortium of sites to very rapidly enroll patients. Even though the population of Australia is much lower than the United States, we can enroll patients there very, very rapidly through these centers of excellence in PDAC. We have, of course, the appropriate regulatory authority to do so, and the program is eligible to receive 48.5% rebate of all R&D dollars spent in Australia. So by doing the studies there, in addition to having more efficiency because enrollment goes faster and a number of the site costs are slightly lower than the United States, we then reduce that by another 50% because of the rebate. It really makes it very, very efficient. And the rebate comes directly from the Australian government, and you apply and receive a check on an annual basis based upon the work you've done. Just need to scroll down here a second. If the Qilu phase 1/2A shows promising results this year, what is the timeline for moving this to phase 2B? Well, I can only project that because our next joint steering committee is later this month. But our expectation is that Qilu will move to phase 2B sometime later this year based upon the results. And while I can't divulge the exact results from the Qilu study because it's been submitted as an abstract for the ASCO meeting in June, and so the results are embargoed at that, I can at least hint that the results seem to corroborate what was seen in Australia. And so we would expect that we'll be moving to further clinical development in China. How do you manage the development of the new clinical relationships in so many different indications worldwide? Well, we do it very carefully and with a very strong communication network. So we have really a network of partnerships where we have regular meetings with folks. We control the development of the protocols, the reporting of the data. We have real-time access to the safety information, and we control all the regulatory situations. So even if the trials are being done under the sponsorship officially of others, it really gives us an opportunity to really manage the communication and control in a very effective real-time way. And the moderator has just told me I'm out of time. So if anyone has further questions, feel free to contact John, and we could always follow up offline. But I want to thank everybody for their interest today, and I look forward to seeing you as investors in Lisata as we move toward completion of development and hopefully a way to better treat cancers of the gastrointestinal tract. Thank you for your time today, and good